It's really important for us.
Okay,
So,
it's a pleasure.
so I think it's time to start. So we are back after our short break with two exciting talks on membrane biology. I would like to welcome Professor Pedro Carvalho in Horizon. Small introduction. Pedro grew up in Portugal, where he studied biochemistry before moving to Boston as a graduate student. After his PhD, Pedro joined Tom Rappaport's lab at Harvard Medical School as a Jane Coffin Childs postdoctoral fellow. It was then when Pedro became interested in the mechanisms of protein quality control at the endoplasmic reticulum and identified the ubiquitin ligase complexes involved in ER-associated protein degradation. After establishing his group in CRG in Barcelona, Pedro was appointed the E.P. Abraham Professorship of Cell Biology at the Sir William Dance School of Pathology at the University of Oxford. Since then, he and his research have made a major contribution in the understanding of biogenesis and quality control of membrane-bound organelles. Pedro, thank you for being here with us today, and we are happy to listen to your talk. The screen is all yours.
Um, Thank you, Katerina, for the kind introduction, for the invitation, and for putting this together. It's really a great pleasure for me to to be here and to share our work with you and with everyone that is attending the meeting. So I'm interested in membrane bound organelles. So how they form, how they acquire their identity. And so, and when I mean identity, I'm talking about mostly their protein and lipid composition. So these protein and lipid composition that is typical to each organelle allow them to have specialized functions. And also, as you can see in this picture, allow them to acquire very characteristic features. So, for example, by looking at this image, you can clearly see the ER that is studied by ribosomes, or for example, the stacked cisterna of Golgi here. So this identity of organelles is really critical for their function. And so, in the how organelles acquire their identity, boils down mainly to two principles. So one is that proteins have to be targeted and sorted to the right organelle. So as you know, most proteins are synthesized by ribosomes in the cytoplasm, and then they need to be targeted to their site where they function, right? So and this is mostly mediated by signals that are embedded in the sequence of the protein that target them to their final destination. And so basically the sorting of all these proteins that is made in the cytoplasm to their final destination, this targeting is really important for to maintaining organelle identity. However, although these sorting mechanisms work very efficiently, they are not 100% accurate. So on top of this sorting layer, there has to be a quality control process that can basically ensure that any mistakes that happen can be quickly corrected. And this is mostly achieved by quality control processes that in most cases lead to protein degradation. So in case a protein ends up in the wrong place in the cell, it doesn't build up. So the cell has the means to basically detect this mistake and quickly revert it by degrading this mislocalized protein. So two other points I want to make here is that, while in this cartoon, these organelles look very sparse and distributed throughout the cell. So from my previous EM picture, you could tell how packed the cells are. So you can see here again, the cells are packed with membrane-bound organelles. So basically, these sorting and quality control systems have to operate in a very crowded environment. So any mistakes that might arise. Hello?
I'm sorry, Pedro, can you hear me?
Yes, I can hear you.
We cannot see how you change your slides if you are. Unfortunately.
They are not moving.
No. So maybe you can try to stop sharing your screen and try to share it one more time.
Oops. Okay. So let's try again.
Yes, now we see the next slide. Maybe you can try. Yes, now it works
Is it
perfectly.
moving? Okay.
Yes,
Okay.
yes.
So I'm
You can
okay. So
you can
so in
continue.
this slide, so I'll go from here. So I was mentioning that I would like to stress 2 points. So the first one is that these sorting and quality control mechanisms have to operate in a very crowded environment of the cells. So they have to be, pretty robust. The second point I would like to stress is that these sorting and quality control processes have to be extremely active in the organelle that I'll be spending most of my talk talking about, and this is the endoplasmic reticulum. So the endoplasmic reticulum, as you can see in the cartoon here, it doesn't receive only the proteins that operate in the ER, but actually it receives all the proteins that are then distributed throughout the secretory pathway, like the Golgi complex, endosomes, and plasma membrane. So basically, all the proteins that enter the ER, they have to be sorted again to be sent to their final destination. And during this process, they are subjected to a very stringent quality control process. So a few things about protein quality control systems. So the first thing is that they are universal. So if you go like bacteria, you have protein quality control systems, and these, evolved in many different flavors into eukaryotic cells, and all the way to man. So in eukaryotes, these quality control systems are actually multi-layered, and some evolved to operate in an organelle-specific manners. Right now, we have protein quality control systems described, for example, in the cytosol, in the nucleus, in the ER, that's the one we'll be talking about, but also in mitochondria or even from plasma membrane proteins. So protein quality control has diversified. And so altogether, these quality control systems, what they do is that they maintain protein homeostasis at the organelle level, at the cellular level, and in metazoa at the organismal level. And so, and defects in quality control systems are usually associated with diseases like, for example, protein aggregation diseases. So they are important both in health and disease. So the quality control system that I'll be talking about throughout my talk is the ER-associated protein degradation, and this is operating in the endoplasmic reticulum, as I mentioned. So from work of many labs, we know that the ER is usually a sequence of events that is described in this slide. So it starts with the recognition of substrates. And so once the substrates have been recognized, they need to be moved across the membrane of the ER. And this step is called retrotranslocation. And in the case of the luminal substrates, the whole substrate needs to move through the membrane, while in the case of membrane substrates, only certain domains need to be retrotranslocated. So on the cytoplasmic side of the ER, they are ubiquitinated. So ubiquitin molecules are covalently attached to the substrate. And this is important to recruit a ubiquitin-dependent ATPase that is called CDC48 in yeast or P97 in mammals. So basically, ubiquitinated substrates recruit these ATPase to the membrane that through rounds of ATP hydrolysis can extract the proteins out of the membrane. And once in the cytosol, a number of chaperones and shuttling factors can then deliver the substrate for proteasome where it is finally degraded. So this sequence of events is basically defining the ERAD, but ERAD is not just. So I should mention, actually, like Katrina mentioned in the introduction, that actually when I was a postdoc, we identified that this sequence of events is basically coordinated by ubiquitin ligase complexes in the membrane of the ER. And we now know that with the diverse sets of ERAD substrates, there's not one, but there's many. For example, in yeast, there are 3 of these E3 ligase complexes, each one with a different substrate specificity. And in mammalians, there's many more, at least a dozen. So and they all have different substrate specificity. We don't fully understand how this is achieved, and actually, during my talk, I'll mention this, a few examples of that. So, but in yeast, there's the hERD, DASI, and the DOA complexes, while in mammalian cells, there's homologs of the hERD and DOA, so we don't have a good sequence-based homolog for the ASI complex, and there's in addition to those a number of other E3 ligase complexes. So what about substrates? So ERAD substrates are very diverse, but probably the best well characterized substrates for ERAD are misfolded proteins. So these are proteins that fail to acquire their native structure. They fail to fold, either because they have a mutation or because they don't have the binding partner available at the time. So basically, they cannot acquire their native conformation, and they are detected by these quality control systems and degraded. Another class of substrates are proteins that end up in the wrong place. So proteins that end up mislocalized in the cell, they can also be recognized, as misfits and by this pathway and get degraded. And finally, there's a third class of substrates, and these are actually folded proteins, but that for some reason the cell doesn't want to keep, so it wants to degrade. So these are folded proteins that at some point need to be degraded because, for example, the metabolic state of the cell needs to be altered, and so the cells need to turn off or on specific pathways. So while these two kinds of substrates are constitutively degraded, in the case of these substrates, their degradation is tuned up depending on the metabolic state of the cell. So, and in my talk, I'll give you two brief examples of these two cases, and at the end, I'll tell you some more about the first type of substrates, misfolded membrane proteins. So what about signal-regulated substrates? So here, I'll give you the case of the sterile biosynthetic pathway. So this is a highly conserved pathway that is essential to produce cholesterol or other sterols for our membranes. And so, we know quite a big deal about this substrate and about this pathway. And a few years back, using a proteomics approach, we found that one enzyme of this pathway that is called erg1 yeast or squalene monooxygenase in mammalian cells was a substrate of the ERAD pathway mediated by the DOA10 complex. And basically, this is a cyclooxamide chase experiment where we're following the turnover of erg1. So what we do is that we block protein synthesis at time 0, and then we follow the fate of the protein over time. And you can see in wild-type cells that erg1 is a quite unstable protein. It has a relatively short half-life of around 60 minutes. So in wild-type cells, erg1, it's degraded very efficiently over time. On the other hand, if you have a defect in the DOA10 complex, like for example, deleted DOA10 or any other recipient of this complex, what happens is that erg1 is no more degraded. So it has a very long half-life. It's a very stable protein. On the other hand, if we delete, if you introduce mutations on the hERG complex, that is another ERAD branch, hERG1 is still degraded with normal kinetics. So these experiment and some others shows that erg1 was actually a substrate of the DOA10 complex. And so there was already the precedent that some of these enzymes were degraded depending on the metabolic state of the cell. So we decided to test whether, manipulating the flux of the sterile biosynthetic pathway, we could change the half-life of erg1. And that turned out to be the case. So for example, if we acutely block the pathway with a small molecule inhibitor by blocking the pathway at this level, at the level of squalene synthetase. So basically, we have an acute stop of squalene synthesis. What happens is that the erg1 stability changes radically. You can see again in wild-type cells, it has a half-life of 1 hour. In cells that have been treated with this inhibitor of squalene synthetase, the half-life is dramatically expanded. Okay, so it seems that the stability of erg1 is responding to the metabolic flux through this pathway. On the other hand, if we do the converse experiment, so if we block the pathway more downstream, for example, at the level of lanosterol demethylase, like using a small molecule called fluconazole, so the cells build up lanosterol, and under these circumstances, what happens is that erg1 is more quickly degraded than in wild-type cells. So this is, again, the time point in minutes, and you can see that after 20 minutes, we barely have any erg1 left in the cell. So in this degradation, is completely dependent on the ERAD branch that is assembled by the DOA10 complex. So cells that are mutated in the DOA10 or any of its accessory factors are completely enabled to degrade ERG1. So from this experiment and the number of other experiments that I'm not showing you today, we show that basically the degradation of ERG1 is mediated by the DOA10 ERAD branch. And this degradation is depending on the levels of lanosterol in the cell. That is a key intermediate in the sterile biosynthetic pathway. So basically, what we showed at the time is that the levels of lanosterol are controlled through a negative feedback loop, through the control of the half-life of the ERG1 in yeast or squalene monoxygenase in mammalian cells. And basically, the enzymes involved were the DOA10 E3 ligase or its mammalian homolog TEP4. So together with prior data from other groups, like for example the Hampton group working in yeast, or Brown and Goldstein or Russell DeBose Boyd in mammalian cells, we now know that another key enzyme in this pathway, HMG-CoA reductase or HMG2 in yeast, is the levels of this enzyme are controlled by a similar negative feedback loop. So basically, together with this earlier work, what our work showed is that ERAD plays a central role in controlling sterile metabolism in cells. And this is mediated by two independent ERAD complexes. And, you know, this dual regulation, actually, if we look at the overall metabolism through this pathway, it makes a lot of sense. So this is speculation, but we think that by having dual regulation of cholesterol biosynthesis, one very early on at the pathway, we can shut down the complete synthesis of sterols. On the other hand, if we block at the level of ERG1 or squalene monoxygenase, we can reduce the synthesis of sterols, but you can still produce a number of other key molecules in the cell that are also products of at least the top part of this pathway. So basically, by having these two control points, you can independently regulate the levels of a bunch of key molecules in the cell. So this was an example of how ERAD can control the stability of a folded protein. So as I mentioned, the ERAD also degrades proteins that are present in the wrong place in the cell. And so I'll mention a couple of cases just as a small vignette. So one of these is the inner nuclear membrane. So the inner nuclear membrane is continuous with the rest of the membrane of the ER. So the ER forms a continuous membrane system, and the inner nuclear membrane is still linked to the remaining of the ER membrane. However, it is a very distinct ER domain. So basically, the first and most obvious difference is that, in contrast to the remaining of the ER, the inner nuclear membrane is not exposed to the cytoplasm. So the inner nuclear membrane is facing the nucleoplasm. And in the nucleus, it's not involved, in the normal typical functions of the ER, like for example, protein biogenesis or lipid synthesis. So the inner nuclear membrane has been implicated primarily in controlling the positioning of chromosomes and how these, for example, have an impact in gene regulation and gene expression. And so these functions of the inner nuclear membrane are carried out by a distinct set of proteins than the ones that localize to the ER that is exposed to the cytoplasm. And so basically you have a continuous membrane system, but that basically has a domain that has a completely distinct set of integral membrane proteins. And initially it was thought that this specialized domain of the ER was basically generated and maintained by selective protein targeting. So for example, proteins in the inner nuclear membrane, they are made in these regions of the ER, but they will diffuse through these lateral pores, that are present in the nucleopore complexes. And once they would reach the inner nuclear membrane, they would interact with nuclear components and they would remain there. And so basically, this would be one way to spatially segregate the proteome in the inner nuclear membrane and the remaining membrane of the ER. So a few years back, what we found is that actually there is this distinct proteome of the inner nuclear membrane is also maintained by selective protein degradation. So we identified E3 ligase complex called the ACI complex that localizes specifically to the inner nuclear membrane. And what it does is that it degrades proteins that are not do not belong to this territory of the ER. And so, this was interesting, and because basically it provided a new mechanism to maintain the identity of this specialized ER domain. So more recently, and actually in collaboration with Alex Steinlab, we show that this ACI complex, this inner nuclear membrane specific E3 ligase, targets these mislocalized proteins by recognizing specific features on their transmembrane domain. So we had a paper that came out earlier this year describing this work. So another way by which protein degradation can basically maintain protein identity was shown in the case of lipid droplets. So lipid droplets is an organelle that forms at the surface of the ER and that it remains in close contact with the ER. And in the next talk from Rashid Tiam, you'll hear a lot more about lipid droplets. So, but a few years back, what we found is that there is a quality control process that also ensures that the membrane of the ER and the surface of lipid droplets have distinct proteins. And this was, again, using a mass spec approach to identify the set of proteins that were initially targeted to the ER, and the end point of these proteins was actually the surface of the lipid droplets. But if these proteins would not quickly accumulate at the surface of lipid droplets, they would get degraded through this DOA10 ERAD branch. And so basically there is some kind of a competitive event. So the protein is targeted, and if it fails to move to the lipid droplet, it quickly gets degraded. And by doing so, you prevent accumulation of a lipid droplet protein in the surface of the ER. So a similar principle was, so our work was in yeast, and a very recent work from James Olsman's lab basically described a similar pathway. So showing that this mode of maintaining organelle identity and compartmentalization of proteins is conserved throughout eukaryotes. So basically, this illustrated, like in very shortly, two modes by which ERAD can affect the function of organelles. And for the remaining of my talk, what I'll be talking about is how ERAD selects misfolded membrane proteins. So as I told you, there's huge diverse sets of misfolded proteins, and there's also a large set of E3 ligases. So who degrades who? Like how does each component reaches its dedicated substrates? And this is, and we think this has been a problem for a while. And I'll just give you a few things, a few slides to illustrate this idea. So when we talk about luminal substrates, so proteins that misfold in the lumen of the ER, it's the route for degradation is relatively straightforward. So they all converge on this HERD1 E3 ligase complex, and this is basically whether we are talking about yeast or mammalian quality control. Whenever we have a misfolded ER luminal protein, it converges on the HERD1 E3 ligase complex. In contrast, if we have a misfolded membrane protein, we are unable to predict today which ERAD branch it will get, it will be engaged with. So it's really, we don't have the set of rules that allow us to understand, what kind of lesion is recognized by each one of these E3 ligases. So this is a question that we are very interested in understanding, because if you want to use, let's say, some predictive power to target specific, let's say, disease-related substrates, we need to have this knowledge. So basically, this problem is complex in yeast because, there are 3 different E3 ligases, but become even more complicated in mammalian cells, where there's at least 27 to 30 E3 ligases in the endoplasmic reticulum. We don't know whether they are all involved in ERAD, but a substantial fraction of these is definitely involved in ERAD, and we still don't understand how each one of these E3 ligases recognizes its substrates. So in order to basically to address this complex question, so we're trying to break it down in a more, in a simpler one. So what we've been doing is that we've been developing relatively simple misfolded membrane proteins and trying to follow their degradation to try to extract these rules of substrate recognition. So until the end of my talk, I'll tell you about two substrates that I think have been quite instructive and for us to start dissecting these rules. And so, and this work is with two proteins, one called ERG11 and one called CYP51 A1. So basically, these two proteins, so they are homologs. This one is from yeast, and this one is from mammalian cells. And there's we know quite a bit about these proteins. There's even a crystal structure of the yeast protein, and it's an enzyme, and the enzymology is well known. So this protein is well studied. But from our, this is mostly work of others. But we found recently that if we make a truncation right after the transmembrane domain, so basically if we get rid of all the cytosolic domain, that is the catalytic domain, this protein cannot fully fold and become a very good erratic substrate. And so we decided to investigate how these, potentially good erratic substrate, at least based on our yeast studies, was degraded in mammalian cells. So we expressed in mammalian cells, the ERG11, that is this truncated ERG11 that is derived from the yeast protein, or we expressed this truncated CYP51 that is derived from the mammalian protein. And we appended a GFP moiety to both of these molecules so that we could basically use the brightness of GFP or the amount of GFP as a readout for the abundance of the whole protein in the cell. So we could use, for example, flow cytometry. And so, and that is shown here. So if we introduce ERG11 in the cell, this truncated version, or CYP51, and if we block, for example, P97, that is these ATPase that is very early, acting early in ERAD, we can see that the GFP increases in the cells, both in the case of ERG11 or CYP51. The similar observation occurs if we block the proteasome. So if we have a proteasome inhibitor, so if we block the last step, again, we have more GFP in the case of ERG11 and more GFP in the case of CYP51. On the other hand, if you block autophagy, that is another major degradative route for proteins in cells, there's really no effect. So basically, based on the dependence on P97, on the dependence on the proteasome, and the fact that both of these truncation proteins are associated with the membrane of BR, we concluded that these are bona fide erratic complexes. So then we wanted to follow up on the mechanism by which they are degraded. And so to do that, we start, knocking out selective erratic components. And we had an interesting surprise. So for example, the ERG11, if we block the TEP4 erad branch, ERG11 is no more degraded. On the other hand, CYP51, that is this, it's based on the homologous protein, but just from the different species. When expressed in the same cells, it doesn't really care. So you knock out TEP4, and CYP51 is still degraded as in wild-type cells. So these results show us that although these two proteins, they are similar and they both engage in ERAD, they were engaging in very different ways, okay? And so this was, TEP4 is an E3 ligase, and so these E3 ligases work with ubiquitin conjugating enzymes. So if you look at the ubiquitin conjugating enzymes, we can knock out the ones that work with TEP4, and again, they affected ERG11, but they did not affect CYP51. So we screened a few additional ubiquitin conjugating enzymes, and we found that UB2K or UB2D3 affected the CYP51, but had no effect on ERG11. So again, confirming that these two substrates were following totally distinct erratic routes. So the cell was actually perceiving these two, recognizing these misfolded proteins in very different ways. So since we didn't have any good, hint of what would be the E3 ligase that was working in the case of CYP51 misfolded protein, we decided to move to a genome-wide screen. So, and nowadays this can be easily done using CRISPR-Cas9 systems. So, and that's what we did. So we did a CRISPR screen to identify the machinery involved in the degradation of CYP51A1. And it was comforting to see that one of the top hits was the UB2K, so the E2 enzyme that we already knew that was involved in CYP51 degradation. And we were looking for the E3s that were involved in the process, and actually we identified two E3s as top hits. So one was called RNF185, and the other one was UB3C. So in addition, we also identified as a very top hit one ER protein that whose function was unknown, but it had very interesting phenotype. For example, in the mouse knockout, this protein is essential for motor neuron function. So if you knock it out, the mouse are still born, but they die soon after, basically because all their motor neurons die. And there was some preliminary data linking this protein to protein quality control. So it was very interesting to that it came out as one of our top hits. So basically, until the end of my talk, I'll be focusing on these proteins, and I'll try to give you some idea how they come together to promote degradation of a subset of membrane proteins. So the first thing that we did was to validate the results of the genome-wide screen. So basically, we use independent guide RNAs to knock out each one of these proteins, and we could confirm the phenotype. And then if we express back in the knockout cells, if we express back the cDNA, for example, for RNF185 or membrane or SPP, that was another hit but in the screen, but I'll not be talking much about this, or UB2K. So expression of the wild-type cDNA in these knockout cells restore the degradation, so the hits were specific. On the other hand, so some of these proteins were enzymes like for example 185 or SPP or EB2K. If we express the catalytically inactive version of that protein, we cannot restore the phenotype. So this suggested that the protein was necessary, and its catalytic activity was also important. So we then tried to characterize more the function of this membrane protein that we didn't really know how it worked. So what we did was we did cyclohexamide chase experiments to see whether the effect on the degradation of CYP51 was at the level of protein turnover. So here is again a cyclohexamide chase experiment, and you can see that in wild-type control cells, the CYP51 is quickly degraded, but if we have knockouts of 185 or membrane, the protein is completely stabilized. And while if we knock out this other E3, UB3C, you do see a stabilization, but it's only partial, right? So if you quantify, you see that UB3C has an intermediate phenotype, while the 185 and membrane are completely defective in degradation. So these two proteins are involved in ubiquitination, so we decided to check whether they were really directly ubiquitinated our substrate, and for that we look at the ubiquitination status of our substrate in different knockout cells. So in parental cells, you see that our substrate is again very actively ubiquitinated, but if they fail if they have deletion of RNF185 or membrane, the ubiquitination is largely disappearing. On the other hand, if we have a deletion on UB3C, we see that the ubiquitination is still there, but the length of the ubiquitin chains it's much shorter. And so, and this is consistent with recent work from Ron Capito's lab that showed that UB3C is involved in mostly extending and generating branched ubiquitin chains. So Basically, what we concluded from this experiment is that UB3C is important for efficient degradation by extending ubiquitin chains, but RNF185 is the main E3 ligase involved in ubiquitination of CYP51A1, and membrane appear to be, involved in the process, but how it would contribute to ubiquitination was not clear. So then, to clarify this, what we did is that we decided to purify membrane, and so we tagged endogenous membrane with a neon green tag, and we immunoprecipitate, and we found that membrane was indeed associated with RNF185, but not with the RNF5, that is a paralog of 185. So it associated specifically with RNF185, and also with two other proteins that had links to ERAD, but whose function was unknown, so Tmod1 and Tmod2. We also use as a control another E3 ligase, so HERD1, and we see that HERD1 interacted with CEL1L, that this was already new, but basically we didn't find common interactors, so it seems that we were looking at the defined complex. But this association with Tmod was interesting because of the prior links to ERAD, and so it allowed we reevaluated our screen data, and we saw that actually Tmod1 and Tmod2 were identified as weak hits in our screen, so they didn't make it at the cutoff, but they were actually out above background. This is interesting because these are two paralog proteins, so we thought that perhaps this is because they can compensate for each other. And so what we decided to do was obvious experiment. So try to knock out both simultaneously, and then we did that. If we make a double Tmod1 and 2 knockout, then we have a very strong stabilization of our substrate. So it seems that Tmods are also important for the degradation of CYP51A1, although only one is sufficient for that. So they have redundant functions in this process. So we also, to further characterize, so they interact with each other. So when, like in ERAD, we have mostly, these units that form complexes. So we thought, characterizing in more detail whether the RNF membrane would define a complex, a ERAD complex. So we use size exclusion chromatography and immunoprecipitation to characterize this further. And we saw that basically both membrane and RNF185, they separated in a single peak, and this peak coincided for both of them. And we also had some substantial fraction of Tmods on this peak, so Tmod1 and Tmod2, and some of the HERD1 was also in this peak. So to confirm that these were independent complexes, what we did is that we took this single fraction, we did immunoprecipitation for membrane again, and we saw that again membrane associates exclusively with RNF185 Tmods, but HERD1 is not there. So basically from here, we concluded that RNF membrane Tmods define a novel ERAD complex. And so here I only show you that this complex is involved, it's implicated in degradation of this truncated misfolded protein, but we actually now identified some additional endogenous substrates for this complex. So here, just to conclude, I want to show you one final experiment. So, we show you that the cell is, has an exquisite specificity in how the different ERAD branches select their substrates, but we still don't understand what the rules for that selection are. So at the end, since we had like two substrates that had, very domain organization, they have very similar domain organization, and and they were derived from this modular architecture. So what we decided to do is try to exploit that to understand what is the features that are involved in each case in the substrate selection. So what we did is that we decided to generate chimeric constructs. So we're starting with the CYP51 truncated protein, and we basically added the amphipathic helix of the ERG11 to this construct, and we observed that when we did that, basically this modular chimeric protein was no more detected by quality control. So the protein is expressed, is targeted to the ER, but it's now fully stable protein. So it's not degraded by quality control systems. So we don't really know why. On the other hand, we had a much more interesting result when we replaced the transmembrane segment. So we again started with the CYP51 construct, but then we replaced the transmembrane segment with the one from the ERG11. And here we observed that the protein was actually extremely unstable, almost pretty much like the original construct. What was surprising is that when we had the CYP51, we knock out RNF185 or membrane, the protein was completely stable. On the other hand, in this chimeric construct, we knock out either 185 or membrane, and the protein was still degraded, just like in current control cells. So what was even more exciting was actually when we then knock out TEP4, the E3 ligase that degrades ERG11, we saw that it got strongly stabilized, and additional mutation of RNF5 had no effect. So basically, what we do, what happened here is that by simply changing the transmembrane domain of CYP51 by the one of ERG11, we completely switched specificity in the degradation of this substrate, and was now the ERG11. This substrate was now degraded by the machinery that recognizes ERG11 substrate. So what this tells us is that TAP4 primarily recognizes its substrates through their transmembrane segment. On the other hand, it also tells us that the RNF membrane complex requires multiple features to select their substrates. So we don't really fully understand what these features are, and this is something that we are very actively looking after now. But basically, it tells that these different erratic complexes basically scan the substrates for different features, and based on specific determinants, then they are selected for degradation or not. So basically, I'll just finish my talk by summarizing some of the data I presented. So I show you that RNF, membrane, and Tmod defined a new erratic complex involving quality control of membrane proteins, so that the recognition of substrates by this complex is complicated and involves multiple features. On the other hand, the TEP4 erratic complex primarily recognizes substrates through their transmembrane segment. So to finish, I would just basically would like to conclude by saying that this protein quality control system in the ER, so that is mediated by ERAD, it's a master regulator of ER organization and homeostasis, and this is achieved by degrading different classes of substrates. So both proteins that are misfolded, so proteins that cannot acquire the right native structure, either because other substrates that do not acquire their final correct localization, or just simply proteins that need to be degraded for the cells to switch their metabolic state. And so in all three cases, degradation of substrates by ERAD is important for the organelle homeostasis organization. So to finish, I would just like to acknowledge the people that did most of the work. So the names here in bold are everyone that contributed to this last part of the work. So the work was led by Mike, shown here, with the help of Loges, that is here, Jake, and Lily. And so Umbretta and Nivedita, so they contributed to earlier work that I went more quickly. So throughout, so our work is funded by several agencies, and we collaborated with a number of people and received reagents from some more. So I'm happy to take any questions that you might have. So I'll stop here. Thank you.
Pedro, thank you very much for the very interesting talk and for guiding us through the different ERAD modes and substrate selection mechanism, as well as the novel ERAD complexes you discovered. So I will just go through the most upvoted questions. The first one is from Florian Keller. So does the altered glycosylation pattern O or N glycosylation of the very same protein influence the degradation mode and speed? if the cell does, for example, no longer need it or somehow accumulates it within a certain compartment? Or is it alternatively rather a question of mature against immature protein?
Yeah, so that's a that's a complicated question to address. So I have to say that so for luminal proteins, so proteins that are in the lumen of the R, the glycosylation state is critical for their ERAD engagement, and it's a major determinant, for example, for engagement on this HERD1 complex that has a very important role in degradation of luminal proteins. So, and this is mostly, end-linked glycosylation. And the way that people think that the way that end glycosylation is linked to quality control is that so the end-linked glycan, it has a complex structure with some glucose mannose residues and acetylglucosamine, and basically these residues the various sugars are trimmed by a number of enzymes in the ER, and when the molecules remain in the lumen of the ER for a long time, and this is indicative of problem of folding. So certain residues or certain combination of residues are exposed, and these basically generate the binding site for some recognition factors through ERAT. So clearly, for luminal proteins, the sugars have a dramatic effect. So and if you actually can express the same protein without the sugars, and it still goes through HERD1, but some of the components involved might differ. So the presence of sugars definitely has a very important role in selection of luminal proteins. So the substrates that we talked about today, none of them is actually a glycoprotein. So they have no sequences for end-linked glycosylation, and they are also not modified by O glycosylation. So basically everything we see here is independent of glycosylation. So yeah, I don't think that glycosylation is a major factor for most membrane proteins, but it might be for some. It's clearly for some, yeah.
So I will move on to the next question.
Okay.
It's my question. So proteins like DGATs contain hydrophobic domains, and recent publications have shown that these can diffuse relatively fast to the lipid droplet monolayer. So do
Mhm.
you think ERAD in this case is a driving force for their localization in the lipid droplet monolayer, or it's rather a clearance system for the leftover mislocalized protein on the ER membrane?
Yeah, I think it's more like the second, right? So I think that Iran. So what we showed in our paper is that basically the IRAD machinery was degrading only the pool that is in the ER and not the one that was moved to the lipid droplet. And we think that that this mostly clears up what is left over, like under normal targeting. But you might imagine that, for example, there might be conditions in the cell in which the lipid droplets actually need to be consumed, and so everything that is at the surface of the lipid droplet somehow is trafficking back to the ER. So in this case, basically these proteins that are trafficking back to the ER are likely to become substrates as well, right? Not only during the biogenesis, but also during, for example, lipolysis and the reverse process of trafficking back to the ER. But actually, there's not a lot known. This is mostly speculation. So, my guess is that that's the mechanism. Yeah.
Thank you. So the next question, it's from Typhoon. Since the different E3 ligases can share substrates or compensate in the absence of other E3 ligases, it is sometimes hard to pinpoint which E3 ligase is actually involved in the ERAD of a specific substrate. Can you please mention some experimental strategies to elucidate the wild type in vivo processes?
Yeah, so it is true. So I think there's good evidence that for some substrates you have some kind of redundancy between E3s. Like for example, for CFTR. So CFTR, you need to actually there's a bunch of different E3s, and they all seem to contribute to degradation of CFTR. In the case of other substrates, like for example the ones I was talking about here today, the specificity is really um, striking, and right there's no crosstalk, so we knock out one E3 and it's like an all or none process, right? So we don't really have I don't have, I don't really know exactly. I would imagine that in the case of the CFTR, that is a multi-domain protein and that has many chances to misfold, the fact that multiple E3s can work on it and can engage it for degradation, it might make sense. Perhaps the fact that these model substrates that we are designing and studying here are relatively short, and basically they cannot expose multiple misfolded features might be biasing to a single pathway, but we don't know. Yeah.
Thank you. So we have one last question because we are running out of time.
Okay.
It's from Claudia. Is CYP51A1 3M an erratic substrate in yeast?
We don't know. We never actually expressed that protein in yeast. Yeah, we don't know. Yeah.
Okay. Okay. Then with this question, I will close our session. Thank you very
Okay.
much
Well, thank you
for
very
being
much.
with
Yeah.
us. And please, everyone, attend the next talk of Rashid. We're going to talk more about lipid droplets. Thank you very much.
Well, thanks. So how do I do to see Rashid's talk?
We will, so you